All right, good evening, everyone. Welcome to the Unit 2 review. This is a student-led review. So how these work is you tell me what you want me to tell you. So just think back on anything in the lecture, video lectures, or the Zoom sessions, or the study guide, or quizzes or labs that you felt confused by or want to review. When you have a question, you just raise your hand and we'll get to it. If we run out of questions, then we have, there are some things I want to talk about with respect to the unknown ID. So I'll let you know when it seems like the popcorn has stopped popping for Unit 2 and open it up for Unit 1 or for unknown ID questions, but also want to sort of review a couple of things about that project. And I also certainly can open the Kahoots for us to do together if you want, if that's something that people want to do. They are also provided as challenges right now. So just let me know by using the raise your hand option if you have questions. If your raise your hand option is not working, you can try posting in the chat. Just know I could possibly miss something in the chat, in which case you might have to post again. So what is on your mind? All right, Tiffany, go ahead.
Could we go over the P-Glow binding cap RNA thingy? I don't know why I have like trouble remembering it. So it's like,
yeah, absolutely. Let me pull up, see if I can find it. I have a slide deck from from lab 6 that, and actually before I show you the slides, I actually want to show you where they are, because in case you don't remember me showing this in lab, I want everyone to know where to find these slides, because it can be helpful in your review. So I'm just going to screen share first my course shell. All right, so in the Canvas course shell, in the lab 6 module, there's this overview page, and on the overview page are these slides. So I'm going to open these up in a second, but I'm just going to show you where they are first. And these slides have all the stuff we talked about in lab with respect to the procedure of transformation, how GFP expression is regulated, how to calculate transformation efficiency, and kind of a taste of what application questions I might ask on an exam. So I'll just switch over now to that slide deck. So what Tiffany was asking is, can we go over how GFP expression is regulated on P-Glow? Why do we have certain predictions? So I'm first going to remind you of the structure of P-Glow and how it works. And then I'm going to go back and review what you're expecting to see on plates. So one of the first genes we need to talk about on P-Glow is this one. This is the antibiotic resistance gene beta-lactamase. If a organism gets the plasmid, then it gets this gene, and this gene makes a defensive weapon against ampicillin. So immediately the cells become resistant to the antibiotic ampicillin. Another gene on this plasmid is the arabinose repressor that regulates the arabinose operon regulatory region that's hooked up to GFP. And the third gene, of course, is GFP itself, which is a green fluorescent protein. And what's important to know is that the antibiotic resistance gene is constitutively expressed, meaning that it is always being made. So if you get this plasmid, you are resistant to ampicillin. And the arabinose repressor is also constitutively expressed, which is important because that means we know that it goes and sits in this operator. But GFP is not constitutively expressed. GFP is regulated, and its expression is regulated depending on environmental carbon sources. This part of the plasmid, the arabinose operon part, is derived from the original arabinose operon. This is the original arabinose operon, and it's got several enzymes for metabolizing the sugar arabinose, an operator that is where the repressor binds and prevents transcription, a promoter that RNA polymerase has a hard time finding on its own, the cap binding site where the activator cap binds to bring RNA polymerase to the promoter. And a couple of important things to remember is if a cell is living in a glucose-only environment, then the absence of arabinose means that the repressor is still in the operator, and we're never going to get transcription of these genes. And furthermore, because we have glucose, there's no cap here, there's no RNA polymerase. When the sugar arabinose arrives, what it does is it causes this repressor to be deactivated, so it never sits down on the operator. And if the only sugar is arabinose, meaning there's no glucose, that means that ATP levels have dropped, cyclic AMP levels rise, cap becomes activated, and it goes and sits in its binding site right here. And that allows RNA polymerase to find this promoter, which is otherwise cryptic. And then we do get transcription. And if both sugars are present, although arabinose removes the repression, the presence of glucose, meaning that ATP levels are high, cyclic AMP levels are low, and cap is not active, so it's not bound here, means that the RNA polymerase is not able to find the promoter. So we don't get transcription for two reasons. For this reason, even though the repressor is gone. So this part here is the part that's been put in front of GFP. So whether or not the cells are able to express this gene and glow green depends on whether or not there's glucose or arabinose or both in the environment. So if we only have glucose and no arabinose, the repressor then is not removed because arabinose is required to remove it. And the presence of glucose means that ATP levels are high, cyclic AMP is low, cap is not active, so it's not here, so there's no RNA polymerase here, so we don't get transcription, so the cells do not glow green, even though they still make this and are therefore resistant to ampicillin, and they still make this, which is sitting right here. If you give them arabinose only, then what happens is the arabinose removes the repressor, so nothing is in the way. In the absence of glucose, ATP levels drop, cyclic AMP levels rise, cap becomes active and sits in its binding site, and that brings RNA polymerase to the promoter, and we do get transcription, the cells glow green. Meanwhile, they're still resistant to ampicillin. And if both sugars are present, then even though the repressor is gone because arabinose is around, the high levels of glucose prevent cap from binding here. And so we don't get RNA polymerase and we don't get transcription. And that's why we have these predictions on these different plates, right? The no DNA control is just making sure we didn't kill all the cells and that they are in fact still sensitive to ampicillin. These are the ones where we actually expose cells to P-glow. And the ones that got the P-glow are all going to live on, and they're going to be selected for, and they're rare, right? Out of millions of cells that are in the reaction, as you can see here, we're only going to get a handful of those cells that actually get the plasmid, and they will survive because they have ampicillin resistance. Then the sugar situation determines whether or not they are green. So in terms of growth, we're going to see colonies on all these plates. In terms of color, we're going to see a glowing on the one that has just the sugar arabinose and not any of the other plates. So Tiffany, let's start with, did that sort of cover it, or do you have specific questions about this that are still kind of bothering you?
No, that makes so much more sense. Like I knew what I was missing to understand it, and now I understand it, like with this visual.
Awesome. Okay.
Thank you.
Now there is another question in the chat that I want to get to, Tao, but before I do that, this is Tiffany's question is a great segue to something I wanted to review with you anyway. Okay, John, thank you for letting me know. So if I go, which direction will I? This one. So this I mentioned in class, but I want to just go over it again. I would expect you to understand how inducible operons are regulated for these non-standard or non-glucose sugars. And so here's another example. And you can use this as sort of a template, like swap all the things out. But I'm going to add one thing into this. Let's see if I can just move this a little bit. So if I said, hey, we're going to use a different plasmid in our transformation reaction. instead of having green fluorescent protein, we're going to have blue fluorescent protein. And instead of being regulated by the arabinose operon, it's going to be regulated by the ribose operon. And instead of ampicillin resistance, the plasmid will have constitutively expressed tetracycline resistance. And what would we expect? So just to sort of give you a visual of what that's going to look like, instead of beta-lactamase, we're going to have a different gene that gives you TET resistance. Instead of the arabinose repressor, this is the ribose repressor. This is where it binds to prevent transcription, and it's constitutively expressed and by default active. Here's the cap binding site where cap will bind if glucose levels are low. Here's the RNA polymerase binding site, the promoter, which is only going to be able to find if cap is here because it's a bad promoter, and it's hooked up to blue fluorescent protein. This is constitutive, this is constitutive, this is regulated. So what conditions do we need to give cells that have been transformed with this plasmid, assuming that they were tetracycline resistant or tetracycline sensitive to begin with? What conditions do we need to provide in order to see this glow? Put that into the chat. And as always, if you have no idea, put a question mark, but do put something. Okay, great. Sarah is right. Just like if we wanted P-glow, GFP on P-glow to be expressed, we need to offer arabinose only. In this situation, we need to offer ribose only. Let's talk about why that is. That's what I thought. I have to go back. The reason we need ribose only, if we want this to be expressed and for the cells to glow blue fluorescent protein would glow blue. If we want this to be expressed, what we need is for there to be transcription, of course. And that's only going to happen if the repressor is gone. This repressor is sensitive to ribose. In the absence of ribose, it's here, blocking transcription. In the presence of ribose, it's gone. So we definitely need ribose to be provided. This also here is a bad promoter. We need cap to bind to the cap binding site in order to bring RNA polymerase here. So we need to remove the repressor with ribose. To get cap here, what do we need to be true in the environment? Yeah, we need cyclic AMP. And how are we going to get high levels of cyclic AMP? Low glucose, exactly. We need low or no glucose so that ATP levels are low, cyclic AMP levels are high, and cap is found. And that will bring RNA polymerase here. And we also need ribose to get rid of the repressor. So we need ribose only, no glucose, basically. So if we did this transformation with the P blue plasmid that I just showed you, and if we assume the cells are sensitive to tetracycline, okay, then what we're expecting on our negative control, just the cells alone on a rich medium with no selection, is a LON, right? And that tells us we didn't kill the cells. We put those same cells on the antibiotic, and what are we going to see? These cells are sensitive to the antibiotic, so what are we expecting to see on the antibiotic of the negative control? No growth, exactly. Yeah. And if I'm interpreting your statement correctly, Tao, you wouldn't see a zone of inhibition because we're not just putting a disc on. I mean, we would if we put a disc, but in this case, this plate has TET all through the medium. So the entire plate is coated with it. The cells are TET sensitive. I know I said TET resistant for a second and I corrected myself, and I'm sorry. The cells are TET sensitive. They're only going to be tetracycline resistant if they get the plasmid, and these cells here in the negative control have not received any DNA. Now let's talk about those cells that did get exposed to the P blue plasmid. Many cells were in the tube, right? We add some DNA, we do the transformation, and we're going to get a few of those millions of cells. You now know from looking at your own plates that few is like maybe 100 out of millions. So we're going to get a little bit. Okay, so when we look at the plate, what are we going to see? Are we going to see lawn, no growth, or something else? In terms of growth, what are we going to see? Colonies, right. Now, and that's because we're putting this plus this DNA exposed or P blue exposed cells onto the selection, right? So they're on these plates that have tetracycline in them. So the only cells that are going to survive are the ones that have received the DNA, which is going to be very few. That's why we would see colonies. This plate only has glucose as a carbon source. It has no ribose. And so are we going to see glowing? That's right. It will not glow because high glucose, low cyclic AMP, CAP's not active, there's no RNA polymerase, we don't get transcription. Now we put the same reaction onto this plate that has tetracycline. So we're selecting for killing everything except the cells that did get the DNA. The cells that got the DNA are alive, they're making colonies, right? So we're still going to see colonies. We're offering ribose and no glucose. Are we going to see glowing, yes or no? Right. And they're going to glow blue because in the absence of glucose, lower ATP, higher cyclic AMP, CAP's active, CAP's bound to the CAP binding site that brings RNA polymerase there. The presence of ribose removes the repressor so we can get transcription. On this plate where we give both sugars, we're going to see colonies, but no glow because glucose is inhibiting the ability of CAP to bind. And so there's no RNA polymerase, even though ribose has removed the repressor. But over time in a longer incubation, we might see some glowing. Why? What happens over time? Glucose could run out, exactly. All right, so those are the slides from there that I kind of wanted to reference. Let me just go back here and to Tao asked if we could review the steps of viral replication. Yes, we can. First, that's a big question. Does anyone have questions about the topic that we just discussed? Before I move on to viral replication. So what I think would be potentially useful to do is go again to the course shell. You know, I drew pictures of the viral life cycles and they're fine. I think they give the right amount of information, but I pointed this out to you guys before. So this might be something some of you would want to have handy. You could print it off for the test, you could refer to it, but what would be far better, in my opinion, if you're taking advice, is that you draw it yourself. Not necessarily that the drawn, if you're a terrible drawer like me, maybe you still use this on the exam, but drawing it yourself will force you to understand it, or force you to realize that you don't understand it, which is almost as useful. So I'm just going to open this puppy up and have a peekaloo at it with you. Okay, so I'm going to go to the DNA life cycle first. There. So we have our virus gets in and the DNA gets inside. The one thing that's not shown here, because this is generic for DNA, is it doesn't show the nucleus. So once this virus uncoats and the DNA is released, this DNA actually has to go into the nucleus. In the nucleus, it's replicated with the DNA polymerase of the host, and it is transcribed by the RNA polymerase of the host to make mRNA. The mRNA is read by the ribosome, and you make all of your genomes and all of your proteins. and then you just reconstitute it. So basically the life cycle of a DNA virus, and maybe if I have my virus one, I can also, yeah, there it is. I'll just also look at that one with you. Let's see, you share that one. If we go down to the life cycle of the DNA virus, again, this is an animal virus, not a bacteriophage. We can look at bacteriophage in a moment. Where is this picture? Oh my goodness. Holy moly. This is very sad for me. Okay. There we go. So here we see a DNA virus getting into the cell, releasing its genome, which is DNA, getting into the nucleus. The host DNA polymerase copies it into new DNAs, extra ones, extra genomes. And the host RNA polymerase makes mRNA. The mRNA has to leave the nucleus and go out and find a ribosome. The ribosome makes proteins out of it. And then you've got all the pieces, the bits and pieces that you need. Let's go back to this. Okay, so that's DNA. The difference between a animal DNA and so this is the wrong picture. I wonder if that would make any sense. Sorry guys, I am a little tired today. My bad. See, this is better. This is the DNA virus. It gets inside the DNA is released. It goes into the nucleus. The nucleus is in the nucleus, the DNA polymerase replicates all of these DNAs. And then the RNA polymerase makes mRNA that leaves into the cytoplasm. And then the ribosomes make all the pieces. And then you have new virus particles. That makes more sense. Sorry about that. If you want to talk about how a bacteriophage does it, it's kind of similar, except there's no nucleus involved, right? So the other difference is, of course, a bacteriophage inject their DNA through the cell wall and they leave the capsid outside. So that's a different scenario. So we can have a quick peek at that in here. Oh dear. Let's have a look at that. This is another thing to remember is that bacteriophage are all DNA viruses. So how bacteriophage work is that they drill a hole through the cell wall, they inject their DNA, and the DNA is then copied by the host cell DNA polymerase, and it is transcribed by the host cell RNA polymerase, and then the RNA is translated by the host cell ribosomes, and we get more virus particles. Now, if we're talking about RNA viruses, the simplest life cycle is the single-stranded positive sense. So here it is. You can see already that this positive sense genome is, in fact, already an mRNA, right? It's at the 5' end, there's the AUG. And if we keep going, we'll find the gene sequences that code for productive proteins. So this gets inside, it takes its code off, the mRNA is released, it goes straight to the ribosome copies it, translates it into capsids, spikes, and the enzyme required to do the replication of the genome. So this RDRP, RNA-dependent RNA polymerase, goes to work copying the genome into a complement and then using the complement to copy it back to positive sense. So now we have lots of pieces to do all of our viral replication work. So then we've got the negative sense, which is much more complicated, but what you can see up front is this genome is not in mRNA already. It's in the 3' to 5' orientation. It's got a cryptic start codon, that's to say it's the complement. And inside the viral capsid, they have to carry an intact RDRP. As soon as it's uncoded, the RDRP then converts this into a positive sense RNA, which can be read by the ribosome to make all the pieces, including more RDRP. RDRPs also change the positive sense that's been made back to the negative senses that it needs to form more virus particles. And double-stranded RNA is very similar to positive sense. Last but not least, the retrovirus life cycle, which is a hot mess. It's very elegant actually, but it is complicated. So you can see that, first of all, this student diagrammed it as enveloped because all retroviruses are enveloped. So they all get in by budding, by fusion. They get in by fusion. And so they get inside, they have to take their coat off. That releases the genome, and these two enzymes, reverse transcriptase and integrase. The first thing that happens in the cytoplasm is that reverse transcriptase changes the RNA into double-stranded DNA. Double-stranded DNA enters the nucleus, integrase integrates it into the host chromosome. And from there, it is transcribed by the host RNA polymerase into the genome again, which doubles as an mRNA, which can leave through a nuclear pore into the cytoplasm and be translated by a ribosome into more reverse transcriptase, more integrase, more capsids, more spikes. And then meanwhile, the process of transcription is churning out these transcripts that are also genomes, which serve double duty, and we get a whole bunch of viral particles. Meanwhile, because it's enveloped, the ER has been busily making these spikes that are sent to the membrane, and as it buds out. So this is slightly inaccurate here. These would actually be all packaged in vesicles because they'd all be made in the ER. They go to the membrane, and as this thing buds out, it buds new spikes out. Okay, so that was fast, but how did that sort of answer your question? Okay. All right, guys. Any other questions? What else is on your mind? What else would you like to know? Ayumi, go ahead.
About the MSL and the MSA media.
Yes.
So like when it's neutron, the color will be red, and acidic, yellow. And how about alkaline?
So we don't normally worry about that because that's not really that informative in terms of the biochemistry. I can tell you what the answer is. The answer is that it would be hot pink, because that particular dye is kind of like a dull red, like almost a brick red or a dull red at pH of 7. As the pH increases, it turns to a kind of almost like a neon pink. But that happens for a variety. It could happen for a variety of reasons. Primarily, it would happen if the organism was unable to ferment the sugar and instead was using a protein source. And so it was releasing a lot of ammonia. But that doesn't really, that's not really substantially different in terms of the question that medium is trying to answer. The medium is used to determine if you can ferment mannitol or not ferment mannitol, assuming that you can survive the high salt in the first place. So the answer would still be no. Red or pink is still a no. Does that make sense?
Okay, thank you.
Any other questions, guys? Haley, go ahead.
Hey, yeah, I don't know if I have a full question, but I'm just feeling overwhelmed by studying the metabolism section. I'm just wondering if you have like a definite resource we should go to. Should we just rewatch lectures, all the videos? Like just filling out the review has been a lot.
Yes. So I don't really have I would say a couple things. So the first place I would try to begin, obviously, is yes, filling out the study guide. I know it's a lot, but I would start there, and I would just pay attention to which things in the study guide do I not understand, or when I write my answer to this question on the study guide, I'm feeling a little uncomfortable with my level of knowledge. Or if someone was to challenge me with this question, I would not be able to to explain it from memory, I would have to look at it and then remind myself of it, because that's going to slow you down. So that's sort of the first stop. But if you're trying to look for sort of hacks, the things that we review in the Zoom lectures are the things that I consider typically more important, I would say. Like they're more likely to be tested on, or I consider them to be a little more challenging, and so I'm spending time on them. So I would say, you know, what parts of the curriculum did we cover in the Zoom might be a way to sort of focus down. And, you know, things on the quiz, but it's kind of everything, honestly. I would say I would check yourself on the quiz questions, making sure you're understanding those quiz questions. I would check yourself on the study guide. I would really pay attention to anything where you run across a question on the study guide and you're like, I have no idea how to answer this, or I would not remember my answer. I don't really understand my own answer. I feel like my answer is incomplete. So that's maybe something to think about right now as you're looking at the study guide that you've created. If there's something on it, maybe there's many things where you're feeling that way about it. I mean, I have the study guide up here and we can go over anything on there that's kind of like feeling problematic. Ayumi says, LB-amp era glue will have almost the same colonies. Yeah, that's a good question. And that's right, Ayumi. You would predict that in a transformation reaction, as long as, let me go back to, I'm going to share my slides again, just because for some people, it's easier to follow when we have something concrete in front of us. Let's go back to, yeah, let's do this. Okay, so here we have the P-glow. So if cells have received this plasmid, then they are resistant to ampicillin. And in the experiment, what we do is we challenge them with ampicillin. We manipulate the sugars in the environment to determine whether or not GFP is expressed. But if they are challenged with ampicillin, then if they have the plasmid, they'll grow. And if they don't have the plasmid, they'll die. And since all of these plates have ampicillin and they're all plated from the same transformation reaction, you would predict that they would have quite similar colony numbers. Now, it is always possible for those numbers to differ by random chance or just making a mistake during plating, but it's not going to be because of the sugars. What the sugars determine is whether or not the colonies will glow. Now, there is something that the sugars also affect, and that is the speed at which cells divide. Okay, so it's not the sugar, glucose, arabinose, whatever, it's not going to affect how many get the plasmid. So it's not going to affect the colony number. But each colony is made from one cell that then divided many, many, many times. So remembering that if a cell is actively metabolizing glucose, it is getting the most ATP, and therefore its generation time will be shorter. Which plate might you predict would have larger colonies? An LB-amp with glucose or LB-amp with arabinose? Which one do you think will have larger colonies? Not different numbers, but larger colonies because a shorter generation time. This is for everybody to think about and put your answer into the chat. And if you have no idea what I'm talking about, put a question mark, but let me know that you're alive. Yeah. And so Ayumi says it was confusing because in lab, there seemed to be more colonies on the one that had AMP plus glucose. And that's just a coincidence. I mean, if you repeated this experiment like 75 times, you would not see that. You would see it more just, so it's just a coincidence. I don't know what else to tell you. And then Tao says LB, yeah, that's right. So the plate that you would predict have the larger colonies would be the one that's glucose. All right, does anyone have questions about transformation, PGLO, anything related to that, or operons, or something else? It doesn't have to be about this. I'll just give you a minute to think, to ponder what parts of the study guide you're feeling sad about, or questions on a quiz, or something we talked about in class. Could be review the steps in bacteriophage lytic cycle in order and naked virus infection in order. Yes. Let's go back. And first, before I do this, I just want to acknowledge that Tao, I know that your connection failed in the middle of that, and you came in kind of at the end. We had gone through all of the life cycles again, and so they're on the recording, but if you have specific questions about some of them, feel free to bring them up as well. Okay, so I had gone through all of these life cycles. I'm just going to review the bacteriophage one, because Tiffany wanted to have a look at that. So here we've got remembering that bacteriophage are always DNA. The phage grabs on to the cell wall. It drills a hole through the cell wall with its lysozyme enzyme, injects the DNA, viral DNA. That viral DNA is copied by the host cell's DNA polymerase to more genomes. It's transcribed by the host cell RNA polymerase to mRNA, and then that mRNA is read by the ribosomes to create capsids. And the assembly process leads to, is followed by more lysozyme being released as the virus is replicated, and that breaks the cell wall open. So that's the lytic cycle. Now, in terms of naked animal viruses, the important part is just to recognize that this isn't like the best picture. Let's see if I can find it. Not there. Not there. There. Is that this is actually the membrane here, and the membrane of the cell is studded with receptors, and the spikes of the virus, that's a naked virus, bind that receptor and triggers an endocytosis event, bringing the whole entire virus particle inside. And then what happens next depends on what type of genome you have, which we just talked about earlier, using the that one student's really awesome diagrams as a guide. So Tiffany, does that answer your question? Do you have something more specific? Okay, great. And then Tao, did you have anything that you wanted to review? Any other questions, guys? And Yumi, go ahead.
Okay, I'm just kind of confusing about constitutive genes is on all the time, but like how different like repressible genes.
Yeah.
I'm just confusing.
I have a couple different slides I want to show. I'm going to go back to this one first, but then we'll look in the actual genetics. So this plasmid has an example of several constitutive genes and then one that's regulated. This is a constitutive gene. A constitutive gene is one where the instructions for making the protein, so if you were to transcribe this into mRNA, the ribosome would read it and make this enzyme, comes directly after a really good promoter. And so there's nothing in between that can prevent the transcription of this. There's no regulation of this. This is an awesome promoter. RNA polymerase always sits down here and then it just transcribes. And so that's all, this type of arrangement, that's always happening. Constitutive means that the promoter is excellent. RNA polymerase can always find it, and it's always sitting down, transcribing, sitting down, transcribing, sitting down, transcribing, as long as the cell is alive. Genes like this that are regulated by a whole bunch of stuff, like in particular, this region called the operator is in between the gene and the promoter. So this is where RNA polymerase should bind. But in between the promoter and the gene, there's an operator. And what can bind here is a repressor protein. And if it's bound, it's going to stop transcription. The difference between inducible and repressible really comes down to what type of repressor you have. If the repressor, as it's being constitutively made, so the repressor is always made, as it's being made, if it's always in the right shape to bind the operator, then it's an inducible promoter, and you need some kind of environmental signal to remove the repressor. If the repressor, when it's made, is made in a shape that can't directly bind the operator, then you need some kind of environmental signal to bind to it and change its shape so that it can and prevent transcription. And so that's how you either get genes that are by default not transcribed because the repressor is always there, or genes that by default are transcribed because the repressor is gone usually. Does that make sense?
Yes, thank you.
Yeah. And the other thing to review, and I'll just very briefly touch base with this, is in the genetics lecture slides, which are right here. I think. Let's see. Yes, they are. You would want to go back and look again at the two kind of poster children for operons, which is tryptophan and lactose. So here is the lactose operon, and here is the repressible tryptophan, right? Which, as you can see, here's the repressor. It's made in a shape that's not normally able to bind the operator, so transcription is just happening. This is a good promoter. Whereas, and then if you want to stop transcription, you have to have something to bind this and turn it into a working repressor. Whereas, you actually go to my this situation, we have a repressor that is immediately active, doesn't need anyone to tell it to sit in the operator, and so it's stopping transcription. To remove this, we need some kind of environmental cue that will bind it, change its shape, and allow transcription. But these types of inducible operons typically actually have a more complex regulation, where in addition to the repressor, which has to be removed if you want transcription, we also have this promoter isn't very good. So actually RNA polymerase can't bind here unless CAP is here, and CAP is sensitive to glucose levels. All right. Stop share. So is there any follow-up questions you have to that, Ayumi, or is that sort of making sense?
Sorry, can I get one more question?
Absolutely.
So regulated genes just make enzymes like amylase or?
Not necessarily. In this course, we've talked about two specific examples. One, a specific example of an inducible operon that regulates the enzymes for metabolizing lactose. And then I told you that's the same way that the enzymes to metabolize ribose and arabinose and all the other sugars are regulated. And we also looked at the specific example of the synthetic enzymes that make tryptophan, and I told you that other things that are synthetic or anabolic enzymes are regulated this way also. But and so that's all you really need to know. But it is true that there could be other things besides enzymes that are regulated in this way that could be inducible or repressible. Let me think if I can think of an example. So remember that there are some species that are halotolerant. Does everyone remember this? Halotolerant meaning that, and halotolerant is different than a halophile. A halophile would be one that like likes living in high salt and maybe even has to live in high salt. But a halotolerant organism is one that can live at normal, so-called normal tonicity of 1%. But if the salt concentration goes way up, like for instance, this Staphylococcus lands on your arm and you're sweaty, it can tolerate and live on the high salt conditions. And so you could imagine that the way it's doing that is via some complex transporter, right? Some protein-based structure that's in the membrane that's pumping salt ions in some particular direction. And if that's the case, then you probably don't want to make that transporter unless you're actually in high salt conditions, right? So the cell would, by default, not make that transporter unless there's high salt. And the high salt would be some kind of inducible signal to remove the repression of a repressor so that we can get transcription of this protein. So it's that protein, that transporter wouldn't be an enzyme, but it's something that has to be environmentally regulated. Does that help? Does that make sense?
Yeah. Thank you so much.
Great. Okay. Tao asked, can we go over in the study guide the question, what is the flow of electrons in respiration, fermentation, photosynthesis, and chemolytic autotrophy? Yes, and let me just share that study guide question so everyone can see it. Okay. So here in the study guide, in the metabolism section, this is where this question is. And so I think this is important, but maybe one of the more abstract concepts. When we talk about metabolism in the past, what I'm guessing is that you have, to some degree or other, had to memorize a series of steps. Maybe you memorize the steps in glycolysis. Maybe not every step, but you kind of memorize that glycolysis is broken down to pyruvate, and pyruvate turns to acetate, and acetate goes to the Krebs cycle, and then we get NADH, and then we have the electron transport chain. And if you get into more higher level biochemistry classes, human biochemistry, you may be required to memorize every enzyme name and every metabolic intermediate, okay? I want you to kind of understand more what is the important thing that's happening in these reactions. And so I focus your attention on electrons and on carbon. As you can see, the next question is, what is the flow of carbon in these same pathways? This is actually an easier question for most people to understand once you know what I'm asking you, but this is, I think, like more important and harder to get. So maybe what I'll do is draw a picture. And I'm going to put this picture in here, metabolism. So give me a second. Screen share. Hopefully this works. Let's find out. It did. How exciting. Okay. So the flow of electrons, let's start with respiration. Okay, and let's do aerobic respiration. The electrons start out in glucose. So here is my glucose with a lot of OHs. So this is glucose, not anatomically correct. Okay, it's a sugar. Everyone can agree that this is a sugar. This is kind of a crappy version of glucose, but I'm too tired to make a real one. So there's the sugar. And in this sugar, there are electrons. That's what we want. So as we break down glucose to pyruvate, one of the things that happens is electrons come out and they're put on to NAD, making NADH. So we've taken some of the electrons out and we've broken the molecule down into pyruvate. So here's pyruvate. It still has electrons in it. So the electrons started out on glucose and then they went to NADH. And also they continued on and there's still electrons in pyruvate. So then what happened? Well, then this was decarboxylated and CO2 was thrown away. And some electrons were removed. We got more electrons out. And that created acetate. So, and acetate still has electrons on it. So we got more electrons out. The flow of electrons is they're coming out and being put onto NADH successively. So there was a bunch of, this is glucose. There was a bunch of electrons in here. We took some of them out. We broke the glucose in half. We made pyruvate, still has electrons in it. We took more electrons out, and now we're down to acetate. And then we go around the Krebs cycle. And what's the Krebs cycle doing? Well, it's throwing CO2s away, but it's taking electrons out and making an NADH. And then those NADHs are going to the electron transport chain, dropping off their electrons, going back to NAD+. And they go through, they get used to make ATP. And where did they end up? Boom, onto water. They end up on oxygen forming water. So they end up in water basically. So if I was to really like super reduce this, I would go, electrons started out in glucose. They went to NADH. And then they went to water. But it's more important they just understand, like, this is kind of a very reductionist view of a respiration. With that idea, do you understand how you would be able to talk about the flow of electrons in, let's say, photosynthesis? Do you want me to go over photosynthesis? Is this enough, or do you want more examples? Okay, so let's do photosynthesis. So here we have a situation where actually the electrons start out in water. Because remember what happens is that the photosystems, whatever membrane they're in, and let's suppose we're in bacteria, so this is the plasma membrane, here's the photosystem. It's stealing electrons off of water and releasing oxygen gas. So the electrons go from water to the photosystem. They're energized by the sun. They flow through the electron transport chain. Their energy is used to make ATP, but then they're actually put back in another photosystem, re-energized, flow through another electron transport chain, and get put onto NADP to form NADPH. So that's where the electrons are now. They started out in water, they end up on NADPH, and eventually where they're going to go is this part here. So ATP synthase is here. Let's say we're getting protons pumped this way, protons coming back this way, ATP is being generated here. What's going to happen, and this is all the light reactions, in the dark reactions in the cytoplasm of bacteria or in the stroma of algae, we're going to have the electrons, which are in NADPH, plus the energy, plus CO2 molecules in the air, all are going to be put together in the Calvin cycle to form a 3-carbon sugar, sort of similar to, but actually more energized than pyruvate. And we get 2 of those, smush them together to make glucose. So if I was to be very super reductionist about this, I would say, well, where did the electrons start in this whole thing? Where were the electrons to begin with, guys? Put that into the chat. Where do they start? Electrons in what molecule are stolen out for this process? Where did the electrons in this photosystem come from? Water, exactly. So we start with water. That's where the electrons are at the beginning. Okay. And then they go to the photosystem, they get energized, they flow through, they go to another photosystem, they flow through. They end up being put onto this carrier NADPH. And then from there, they end up being put onto carbon dioxide, making sugar. Does that sort of help? You know, there's not just one way to answer this question. It's more of a, do you understand basically where the electrons are going? Where did they start? Where do they end up? What happens to them in between? Let's do one more. Now, there's two processes left to talk about, fermentation and chemolytic autotrophy. So I'm going to do one of these without telling you what it is, and you're going to tell me which one I'm looking at. So let's suppose we say, okay, the electrons started out on a metal ion. And then they went through an electron transport chain. They ended up on NADH. They went through, they went to an electron transport chain. And then they went on to water. What process are we describing here? Where the electrons started out in metal, they go through an electron transport chain, and they end up in water. Is that fermentation or chemolytic autotrophy? Like, you all can recognize that, right? If we wanted to, this is obviously chemolytic autotrophy. If we want to describe fermentation, what we would say is, where did the electrons start? They started out in glucose, and then they ended up temporarily on NADH, but they didn't stay there. At the end of the day, they ended up on some kind of organic acid, maybe lactic acid, maybe ethanol fermentation. Tao, does that answer your question, or do you have a follow-up? Good. Okay, I'm just acknowledging that it's 6 o'clock. Let me kind of read the room. Do you guys have more questions about Unit 2 that you want to talk about? Yes or no? Just put your answer into the chat. And just answer yourself personally. You don't have to worry about everybody else. But I will wait to hear from everybody. Great. So I have a few things that I want to talk about with the unknown ID. Tao, you do have a question about Unit 2, though. Go ahead. What's your question? Is the test about the same amount of questions as the last one? Great question. It is very similar. I would say the format of the beginning questions being mostly fact-based true falses, worth 2 points, a short answer question where you reflect on those questions, a section of multiple choice and that are also worth 2 points, that are sort of starting to get a little bit more medium difficulty, then some longer questions like matching and multiple drop down and things. Towards the end, there's the hardest questions that are the longest and require the most thinking and that are worth the most points. Any other questions about Unit 2, guys? John says, can you please personally rate this test difficulty from 1 to 10? I don't know how difficult it will be for you personally. The reason is that I've seen everything. Like I've seen people do poorly on test 1 and well on test 2. Well on test 1, poorly on test 2. The same, both tests were terrible, both tests were great. Okay, so I've seen every possible combination. But if I was to average the average student's experience, this is a harder test than test 1. Yes, Nikki, you get to bring notes again. So you can bring your textbook, you can bring hard copy notes. You cannot bring digital notes unless they're on a device that is not connected to the internet at the time of the test, and you show me that. You can bring a calculator. You're encouraged to bring a calculator. So yeah. Other questions? How much math is it? Well, there's probably going to be some transformation efficiency, growth calculations. What other math did we do? Plate counts. I would say it's maybe similar-ish in terms of the math to the first test. Maybe there's a little more because I feel like we did a little bit more. But I don't feel like the first test was like heavily math based. So the second one might be a little bit more, but it's, you know, it's not going to be all math. There's a lot more to talk about than that. I will say that there's, you know, just like before, there are critical thinking questions. There are reasoning questions where it's not sufficient to know one fact. You have to know 3 facts and then you've got to put them together. So that's for those harder questions at the end, as you remember from last time. Any other questions about this? Okay, so I have another question for you guys then. I have a little bit that I want to say about the unknown ID project, and then I want to open it up for questions about unknown ID. Do you guys want to blow through and get that done? Or, you know, it could go for 15, 20 minutes or something like that, could be longer. And we've already been talking for an hour. So do we need a little break before we do that? Like what's our desire here? We're a small enough group that we can vote on this in majority rules. Do you want a small break and then talk about unknown ID? And if you're not interested in unknown ID, you can like, you can leave. Okay, so we have, so far we've got a like preponderance of keep going and then one person asking for a break. Keep going, then leave. Okay, how about this? Let's take a really short break. I need to check on my family. Just like 5 minutes, like we'll come back at 6:10. So 5-minute break, super short, and we'll see you in 5 minutes and then we'll get it done. All right, guys. So I did want to say a couple things about the unknown ID. I'll cover this in lab as well, but I just wanted to kind of remind you of what the expectations were at this point and kind of the timeline of this project. So I want to share the schedule. See if I can find the schedule. There it is. No, there we go. So it's not like urgent tomorrow, but it is, you do want to be planning ahead for this deadline. The unknown ID lab report deadline is June 5th. So ideally, you want to have identified your unknown by this weekend, but that by the 29th, which is this Sunday, like, so, in 6 days. That is not like a hard and fast deadline. It's just a recommendation so that you have, a week pretty much to write this lab report. Now, it might take you a bit longer, and that's not a problem as long as you've planned ahead, and you can start writing this lab report like right now. You don't have to wait till you know your ID. So I wanted to walk you through some of the components of this lab report so you can kind of see where you can make a start and start to feel like you have made progress, even if you're not finished, and take a little bit of that stress off. So let me show you where the instructions are. Make sure that I have put my recording back on. Yep, I did. Oh man, my internet is being really crazy slow today. Hello. While we wait for my super slow internet, does anyone have questions that came up for them about Unit 2 over the break? That 5 minutes give you a chance to think about something that you desperately want to touch base on. This is making me super nervous. So just so that I know that this is not frozen, if you can hear me, put yes into the chat. Excellent. Okay, thank goodness. Let me just stop and refresh that. Why this is being a pain, I do not know. Go away. Let's go away. Again. There we go. Okay. So, one other thing I wanted to mention. So in week 7, these Kahoots, these are challenge Kahoots. You can use them and you should be able to go and put in your pin and it should work. Okay. Let's go to the instructions for the lab report. Okay. So this kind of walks you through what to do, but you need a title page. You could create that tonight, right? It needs a title and your name and your unknown ID number. The results section, which is just going to have this as its heading, looks like this, and it's going to be longer because you're going to have more than 5 things. But you start with the gram stain. What date did you observe that? Here were your observations. Whatever your initial tests were to get to your group, what date did you observe those? What were your observations? And then all of the subsequent tests that you did. So what appears in this is everything that you ordered that actually had a result, including anything that came back inconclusive, where the technician said inconclusive. The things that don't make it onto this table are going to be anything that you requested and the technician said that's not a real test. For example, many of you asked for oxygen preference, which is not a test. You may have ordered thioglycolate, right? Subsequently, thioglycolate goes on here, but anything you asked for and the technician was like, I don't know what you're asking for, leave it out. But if let's say you asked for a coagulase test or a phenol red mannitol test and she wrote inconclusive, that's a legit test and it goes on here and you just write inconclusive in the observations. So you can start this right now, right? You have some tests, you can put them in, you could get this table out of the way. And then just if you're not done yet, as you do each test, you just add a line item to your growing table. The discussion will have the discussion header, and then it'll have a flowchart graphic. And the flowchart graphic will look like something like this or something like this. These are alternatives. They are not both required. But you're going to start with whatever your group is, and you're just going to, in chronological order, you're going to say, what was the first test requested? What were the species eliminated? Next test, what species were eliminated? Next test, what species were eliminated? The tests that show up here are going to be anything where you got a picture. So you would again leave out anything that you asked for that doesn't exist, like oxygen preference, which is not a test. And you would also leave out anything that was inconclusive because that didn't eliminate anything, but that's not your fault. If, however, you got a picture, then you have to declare it And then if for some reason you made a mistake in ordering your tests and you ordered something that didn't eliminate anything, you would say, you know, whatever, no eliminations. Tiffany, you have a question?
Yes. Do we have to do both of these, or is it one or the other?
One or the other, and it doesn't have to be this exact format either. These are just the two most common. You can use little arrows if you like arrows, or you can use a table. This is the same information, right? So you just choose whatever you want to make. And just make sure it's complete, except for you don't put in the inconclusives, and you have to go step by step. The only difference or the only exception to going step by step is for Enterobacteriaceae, you ordered in the InVic series first, so you can lump them together, but after that, every test has to be on its own. What did this test eliminate? What did that test eliminate? What did that test eliminate? For the Bacillus group, if you ordered multiple pHs or multiple salts or multiple temperatures at the same time, you can put two of them together. And if you're not sure, ask me. But what you'll notice is you're declaring all the species names. Something to know, because that for some groups can be a lot of species names, is that when you look in the Burgess module, there usually are Word documents with all the species listed, and you can just copy paste those in instead of having to handwrite everything. After that, you're going to write about your elimination process. You're going to have a paragraph describing how you got to your group. So here's an example of that paragraph. The unknown turned out to be gram-negative, and then you have to interpret what this is. Notice that I'm not providing any color or observation information. I'm just saying the unknown was gram-negative, and then I'm saying, what does that result mean biologically? Tests X and Y were positive and negative respectively, indicating whatever this indicates biologically, and that shows that it belongs to whatever the group is. After that, you're going to walk through what all these other test results mean in terms of the biology of the organism. This organism was positive for X, Y, and Z sugar fermentations, meaning it can ferment these sugars. It was negative for la 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 exoenzymes, meaning it's not able to metabolize yada yada. And then there's a bunch of guidance around what to do there. Once you know your ID, you're going to say at the end of all that, this is what the unknown ID was. And then you're going to have a 3/4 page section on research, which is based on stuff you look up. So you're going to go out and find at least 3, minimum 3, legitimate peer-reviewed references and cite them. You are also allowed to cite the Burgess manual if you use it. And I'll show you how you would use that. So again, if you go out to Burgess, you will notice this is what I was talking about for some of your groups that have lots of names. You'll see that there's a document that has, oh, that's the wrong thing. Whoopsie doodle. It's the list of names is there. I just didn't click it. This is a list of all the species names. You could copy and paste those. Bacillus is the other really large group, and they have numbers, so you're welcome to use their numbers instead. When you look at Bacillus table, you can see that the species are numbered. And that's true for Staphylococcus as well. So you could use these numbers if you don't want to write out the whole species name in your flowchart. But in your instructions, oh shoot, for the research section, if you want to cite the Burgess manual, this is the part you would cite. So this is all of these tables here, all of them come from the same source, which is the Burgess manual, which is a multi-volume manual. But I've excerpted pages from it that you might want to read. And the reason you'd want to read these is for your research section, once you know what you have. When you go through these pages towards the end, you'll find, will this help us? Let's find out. Oh, you will find a section for every species where every species is described. And I'm trying to find it in here. I wonder if it's in the other one. It is in the other one. Please. you'll find it here. So if you happen to be Citrobacter something or other, and you go through these pages, you'll find that it lists every species. There's a little blurb. I don't know if you can see this because it's kind of sideways. Let's see if I can make it a little more obvious. Here it lists every single Citrobacter and it has a little blurb about it. So let's suppose you ended up identifying Citrobacter galeni, and you wanted to cite this information that it's found in human stool, human urine, human blood, and in the environment. You could say that, and you could cite your source, which is the Birdie's Manual. And the source to use is right here for your bibliography. Okay, so going back to those instructions I was talking about. You can pretty much do all of this except for the research section right now. And again, you won't be finished this necessarily, and you won't be finished that necessarily, but you can get a lot of it done so that if your species ID isn't made until like, Wednesday of next week, you don't have to panic. You have a little bit of time still, and you will have done this or started it, and so you won't be doing everything last minute. Let me go back to the schedule and point something out. So this is when the lab report is due, on the 5th of June. What you'll notice is this whole week we don't have a Zoom class on Monday because of the holiday, and we don't have a lab that week. So you have a little bit of extra time to be devoted to writing. That's all I wanted to say about that. I do want to remind you a couple things. One is that tomorrow we are covering, let me go back to the schedule. Tomorrow night in Zoom, we are covering pathogenesis. So you do want to read and prepare as usual for this topic. And then on Wednesday in lab, we're going to follow up on lab 7. So we're going to actually collect our data. And then that's going to leave us plenty of time to go on up to the computer lab and do the exam on the computer in the computer lab, just like last time. And let me just warn you that if we are done lab early, we will go up to the computer lab a little bit early. So let's suppose that you were caught in a horrifying traffic and you didn't make it to lab until 6:30, and then there was nobody there. Read the board, because I will have left a note in that case that we're upstairs in the computer lab, and that's where you should join us. And you should also not like cry or panic, because the plates that you need to analyze can be saved in the refrigerator for you. Nikki asked, no Zoom on May 30th and no lab on June 1st. That is correct. So next week we won't meet in Zoom on Monday. We'll just meet on Tuesday, and we won't meet in lab on Wednesday. Okay, so that's all I wanted to share with you guys. And if you have questions about your unknown ID progress and you want to talk about that, then stick around and ask. Or if you have other questions about the Unit 2 review that have come up for you while we've been chatting, then stick around. But if you're done, sign out, have a good Monday night, and I'll see you tomorrow.